So, can you hear me now?
Yes.
Okay, good. Sorry about that. Hi again. Once again, thank you for joining us. We are going to listen to Dr. Akbarzadeh. She's a postdoc researcher in organoid technology at Erasmus MC, Netherlands. She's a PhD. She has a PhD in clinical biotechnology from Tabriz University of Medical Sciences, and she's working on modeling and studying the molecular mechanisms of bladder cancer in patient-derived cancer organoids. She's also interested in trying to establish transgenic healthy organoids and bladder tumoroid line using lentivirus transduction and CRISPR-Cas9, which is super interesting. And hopefully she can talk a little more about that as well. All right, so without any further ado, I'll ask her to start her presentation. I think, Maryam, I think you have access to your desktop, so you can start already. I'll take my video off, so this way we don't occupy much of the bandwidth.
Yes, but I want to share. Yeah. Okay.
All right. So I had put you as a presenter, but now I have given you access to your webcam and your desktop sharing. You're not in Iran, are you? Are you in Netherlands?
Yeah, I'm
Okay,
in
good.
Netherlands.
How's the weather?
Weather is rainy.
Oh, okay.
Yeah.
All right. Go ahead. Continue.
Okay, do you have my slides now?
Yes, we do. We have the presenter's view. We don't have your full slide view.
Okay. So, 1 sec. I should change.
So while you're getting your presentation up, I should let everyone know that every month on a Sunday of every month, we have a journal club like this or a presenter like this that will present something of an interest to our School of Advanced Technologies in Medicine, and I invite everyone to stay tuned for every month. All right. Okay, and now we have your desktop.
Yeah. 1 sec. Yeah. Is it
Yeah.
true now?
Yeah, that's good.
Okay.
You can also try to take your video off if you like. Either way, it's okay.
Yeah. So it's better.
All right, take it away.
Okay. Again, I think I don't access to share my presentation. Okay. Okay. Yeah, it's okay. Yeah, it's okay.
Yes, it is. Go ahead.
Okay. Good afternoon, everybody. Thank you very much for your introduction. Today, I'm going to give you an overview about organoid technology. But I don't know why my computer is very slow. Sorry, 1 sec. Okay. Today I'm going to give you an overview about organoid technology, fundamental and application of this technology with focus on bladder organoids. Please don't hesitate to interrupt me during presentation if you have any question and if something isn't clear for you. Organoid model is three-dimensional cell culture systems that closely resemble the in vivo organ or tissue from which they were derived. These three-dimensional systems reproduce the complex spatial morphology of a differentiated epithelium to allow biologically relevant cell-to-cell and cell-to-matrix interactions. They are made of stem cell or progenitor cells and self-assemble when given a scaffolding extracellular environment. Why researchers became interested in three-dimensional cultures? As all of you know, researchers use different type of model for studying about disease. There are some limitation on each models. For example, they work on cell lines, iPS-derived cells, primary human culture, or animal models, but there are some limitation on each of these models. Cell lines are genetically unstable because of interaction with plastic, and then they are not representative for human cells and tissue. Using iPS cells also is not completely useful because of epigenetic changes and instability in this system. The differentiation, loss of function, necrosis, and limitation in expansion happen in the primary human culture system. That's why using primary human culture is not good enough for modeling of some of diseases. Animal model also have some limitation. They aren't representative for human cells. This model is expensive and we have ethical tissue. Organoids are a novel tool for disease modeling. Organoids are composed of ideally all differentiated cells, cell types of a particular organ. This makes them an attractive tool for study of infectious, genetic, and or cancer disease. If you want to talk about the history of organoid culture, we can back to 1907 when Henry Wilson tried to culture sponge cells for a first attempt of in vitro organism regeneration. A few decades later, several groups performed dissociation reaggregation experiments to generate different type of organs from dissociated amphibian embryonic kidney and chick embryos. In 1987, scientists began to improve cell culture conditions by simulating the in vivo microenvironment. Lee et al. Demonstrated that breast epithelial can form three-dimensional ducts and lumen when grown on extracellular matrix. Interestingly, they found these cells are able to synthesize and secrete milk proteins, but when they grown these cells on two-dimensional culture, they couldn't find these proteins. So they highlight the importance of cell-to-matrix interaction in tissue maintenance and differentiation. Organoid research began to shift from two-dimensional to three-dimensional when a group was able to generate cerebral cortex tissue from embryonic stem cell using the three-dimensional aggregation culture method. A revolution in organoid culture system began with the discovery of LGR5 receptors as an adult stem cell marker by Hans Kilovers in 2007. He found LGR5 positive cells can form three-dimensional intestinal organoids in matrigel that self-organize and differentiate into cryptobulous structures in the absence of a mesenchymal niche. This was the first report on establishing three-dimensional organoid culture derived from a single adult stem cell, which set the scene for many subsequent organoid works in other systems, including liver, pancreas, lung, kidney, etc., using either adult stem cell or progenitor cells. As you nicely see here, when LGR5 positive stem cells even start with single cell, can grow and divide in different days, and approximately after 2 weeks can form a structure that have polarized epithelial layers. Basal membrane in the outside, you can see here, and apical membrane toward the inside of the lumen. Interestingly, these organoids also had barrier function. So LGR5 stem cell derived organoids recapitulate organ function and heterogeneity. After this discovery, scientists started to establish organoid culture system in the other tissue as well. I can give you some example of these cultures in the other tissue here. To establish pancreas culture, Clever's group isolated pancreatic duct fragments from adult healthy mice and embedded them in matrigel as a extracellular matrix, and they cultured these fragments with media that containing with different growth factors such as EGF, R-spondin, NAGIN, FGF10. As you see here, after several days, the cystic structures started folding and budding, exactly like the origin tissue. Using this culture condition, they have been able to expand the cultures by passaging weekly for over 10 months. Another organoid type that Clever's group established is lung organoid. Like pancreatic organoid, they cultured airway cells in defined media and extracellular matrix. Under optimized condition, three-dimensional organoids formed within several days, and they could passage and expand them. Based on immunostaining, they found airway organoids were composed of polarized airway epithelium containing basal, secretory, and multiciliated cells. You can see here basal cells, and you can see here ciliated cells, and also secretory cells. It is very interesting that you can see whirling mucus in the in this type of organoid, and if you zoom in, you can also realize beating of cilia. This showed that this type of organoid also, like the other one, recapitulate function of lung system. For development of liver organoid, they again optimized media for liver organoid culture, and they could culture them for several months. Then they tried to differentiate liver organoid by adding or removing some growth factors in the media. As you see here, they could differentiate liver organoid when they cultured in differentiation media. We can see the organoid has organoid acquired hepatocyte phenotype. we can detect albumin expression in the differentiated liver organoid. Gene expression profile also revealed revealed a high level of hepatocyte markers such as albumin, such as cytochromes, and such as apolipoproteins. Okay, now we have different type of organoids. So the question is how we can use them? What's the application of them? One of the most important application of organoid is biobanking. After culturing of organoids from different patient and healthy donors, these mini organs can be stored in biobanks and used for fundamental research or precision medicine and or regenerative medicine. For example, cerebral organoid can be used to understand brain development, and mini guts can serve as a personalized drug testing tool for cystic fibrosis. Mini livers also could form a complement to current organ transplantation to restore liver function of patients with metabolic liver disease. Organoids also can be used as a model for different types of diseases. For example, we can use for a study about cystic fibrosis. We can use for a study about different type of the cancers or infectious diseases. Another application of organoid is drug screening. We can use organoid in drug testing for discovering new drugs and personalized medicine. Several organoid biobanks of diseased and healthy control tissue cover the range of genetic mutation known from a large-scale sequencing analysis, and thus provide the ideal material to screen for new drugs. Numerous studies have also reported the successful use of organoids to predict therapy response in cancer patients, which named personalized medicine. Organoids are currently used to study host cell interaction with microorganisms, including virus, bacteria, parasites. For example, our lab use liver organoids for studying about HIV and HPV infection, or for studying about coronavirus that has overwhelmed the entire world these days. We can use this model. Identification molecular target for drug development also is possible with transcriptome, proteome, and metabolome analysis in organoids. Recently, genome editing based on the bacterial defense mechanism CRISPR-Cas9 has emerged as an easily applicable and reliable laboratory tool. Combining organoids and CRISPR-Cas9 creates exciting new opportunity to study organ development and human disease in vitro. Overall, more important application of organoids is using them for personalized medicine. For personalized medicine application, taking disease and healthy tissue from the same patients as a control allows the expansion and development of organoids line with same characterization and mutation in the lab. Considering the similarity of gene mutation of organoids organoid with their parental tissues, we have possibility to test different drugs based on different mutation in each patient, and that's why the organoids culture is bench to bedside. Organoids provide a promising tool to advance personalized medicine and next generation drug screening and to limit the need for animal experimentation. Okay, now I want to talk about organoid culture methodology. How we can culture tissue as organoids. After resection, normal and tumor tissue from same patient, we should remove fat or muscle section to enrich epithelial section. Then we can mince them and start to digestion. We have to use different type of enzyme based on type of tissue. And after resuspending solution, we can pass cells from 70 to 100 micrometer cell strainer to remove big and undigested tissue. After centrifuging, we can culture extracted cells in mix of BME or matrigel as extracellular matrix in pre-warmed plates. Single cells can grow as an organoid if we add suitable media, if we add proper media to maintain stemness. Every 2 to 3 days, we have to change the media, and when the size of the organoid is big enough, we can digest them and we can passage them and expand the line of organoid. One of the most important section in culture of organoid is defining media. It completely depends on origin of tissue and its organogenesis. As you know, during gastrulation, pluripotent stem cell differentiate into germ layers, 3 germ layers, ectoderm, mesoderm, and endoderm. Cells in these 3 layers will give rise to different parts of the organism. These differentiation processes are affected by stimulating some important signaling pathway with growth factors. For example, for liver, FGF2 and BMP4 are very important for differentiation of the liver to of the stem cells to liver organoid. Or, for example, for kidney, we need FGF9 and Wnt to have a kidney organoid. Wnt is a growth factor that it is very important in the organoid culture because this growth factor can keep stemness in the organoid and they can proliferate and grow as a organoid in structure. After successful culture of organoid, we should confirm that these organoids are similar to original tissue or not. Organoids and original tissue should be similar to each other based on histological and genetic mutation. And normally we use H&E staining to show that our organoids are similar to tissue based on the cell morphology. In most of the organoids that till now is established, they are similar to each other, and we couldn't find any big difference between the histology of the organoid and original tissue. On the other hand, we should check the mutation of the organoid and original tissue. They should be same. We can do sequencing and tumor tissue and organoid, and also copy number variation. Based on our information, we don't have difference between organoid and their original tissue about the mutation and difference in copy number. What I did at Erasmus MC. Our team at Erasmus MC consists of clinician, surgeon, master, PhD, and postdoc researchers that work on bladder cancer. Why we are working on bladder cancer and why we started to establish bladder organoid, these are the questions that I want to respond in next slides. Bladder cancer is the 9th most common cancer in the world, and it has a high rate of recurrence. There is two important type of the bladder cancer. One of them non-muscle invasive bladder cancer, that it is low grade of bladder cancer, and muscle invasive bladder cancer, that cancer cells start to invade to muscle section of bladder, and it is high grade of bladder cancer. There are different types of treatment for bladder cancers. TURBT is endoscopic resection. It has diagnostic and therapeutic role in low grade of cancer. Cystectomy is surgical removal of all or part of the bladder that normally used for high grade of this cancer, and neoadjuvant and adjuvant chemotherapy with different chemotherapy agents like mitomycin, doxorubicin, cisplatin, and gemcitabine. That our purpose with this therapy is shrinking a tumor or stopping the spread of cancer and or killing any remaining cancer. Despite extensive treatment methods, approximately 50% of patients will die within 5 years due to progressive disease. So we need to find out how we can target effectively bladder cancer cells and how we can improve overall survival of patients. To reach this goal, we needed a good model, which should be much more similar to human bladder, genetically and histomorphologically. So we decided to establish and optimize a patient-derived bladder organoids and tumoroids. As I explained in detail in the previous slides, after tissue processing and cell isolation, the isolated cells were seeded into BME as an extracellular matrix. Then we tried to optimize the bladder organoid expansion media by adding some or removing some tissue-specific growth factors and proliferative factors. As you can see here, the single cell are seeded in BME, and after a couple of weeks, you can detect the circle-like structure that these are bladder organoids that we can passage them and expand them. Of course, we should confirm that our organoid is similar to tissue. We also like another study. We did the H&E staining on our organoid, and we compared the result to H&E staining of the tissue and our organoid based on the morphology and histology were similar to original tissue. And interestingly, the organoid cells, our organoid cells can produce mucin you can see the blue sections like the normal tissue, and it means that our organoid also can recapturate the function of bladder tissue. We also check the genetic stability with snapshot analysis. As you see here, all the mutations are similar to each other, so we didn't have any problem, and our organoid were stable based on the genetic mutation, and also copy number variation also was similar to each other. On the other hand, our organoid have different type of the cells, undifferentiated cell, semi-differentiated cell, and fully differentiated cells. You can see here undifferentiated cell, that this undifferentiated cell is around of the organoid, and fully differentiated cells is in the middle of the organoid. This means that we have heterogeneous structure that can resemble tissue structure. Okay, till now we have cultured around 150 patient materials and we biobank them. Our culture efficacy is around 80%, that it is 2 times higher than other culture. After this establishment, we have a bunch of organoids with different characteristics, with different mutations, and also we access to all of the clinical information that then we can use them for a different application. Okay. We are working on different projects now in our lab, and I want to shortly introduce them. We are trying to generate patient-derived bladder organoid from urine stem cells. This can be a new non-invasive approach for in vivo urological tissue repair or in vitro bladder modeling. Also, based on transcriptomic and proteomic information, we can identify new molecular target for drug treatment of bladder cancer. On the other hand, we are co-culturing bladder organoid not only with immune cells, but also with different type of organoids to investigate invasion and metastasis mechanism and finding new therapeutic approach. Our group also are working on chromatin remodelers. Chromatin remodelers, especially BAF complex, is one of the most important complex in bladder. ARID1A is one of the subunit of this complex, which has a mutation in around 25% of bladder cancer patients. We are trying to target ARID1A with some specific inhibitors. For this reason, we established ARID1A knockdown organoid with shRNA. So we have knockdown models for study about and find good target for targeting ARID1A and finding a way to treat, to have a good treatment for bladder cancer. And also we are looking for finding novel therapeutic for GC or cisplatin gemcitabine resistant bladder cancer, FGFR3 mutant, and ARID1A mutant bladder cancer. At the end, I deeply appreciate all of my colleagues, especially my supervisors, Tokame and Telita, that helped me a lot in this way. And thank you for your attention, and it's my pleasure to answer your questions.
All right. Thank you very much for this informative and interesting presentation. Indeed, the organoid technology is fascinating, and we are surely very interested in our departments ranging from biotechnology, molecular sciences, neurosciences, tissue engineering, applied cell sciences, and of course, nanotechnology. I'm sure everyone enjoyed this lecture-like presentation. Personally, I'm super interested to do research in this area and use microfluidics to culture organoids. Before we go over possible questions, I see there's a question. I would ask you to please send me your slides so I can share them with my colleagues. I think some people had difficulty reading some of your slides. So there is one question from Nassim from Dr. Wusuqi. She says, how do the cells shape into a ball-like shape to form an organoid?
Can you please repeat the question?
Do you? It's the last comment in the comment section. So they're trying. So she's asking, how does the cells go around and form a ball?
Okay, yeah. Uh, this is because of the interaction with the extracellular matrix. When we put the cells as a stem cell in the extracellular matrix, their interaction with the extracellular matrix cause that we have this shape of the organoid. They grow and they can differentiate. When they differentiate, they can come in the middle of the organoid, and in the outside we have the basal cells, and most of the basal cells in the tissue also are intact with the membrane, and then its cause may be the shape of the organoid. And also it's not in most of the organoids, all of them is not the round shape. It completely depend and it completely different based on the tissue that we are working and patients. For example, I had one patient that was grape shaped. It was not round because of his mutation. It completely depends on this patient mutation. And or for example, for pancreas, as I showed in my slides, it's not completely round, and this structure just resemble the structure of the tissue. When you have when the bladder is round shape, the cells also try to have a round shape.
A round structure. Okay.
Yeah, round structure.
One more question. They're talking about what are the culture media and growth factors that you're using in order to avoid limitation of subcolonial culture.
Yeah, this is very, very important, and this section is very hard to do. I explained that it's completely depend on organogenesis. You should, when we are working and when we are establishing the media for organoid culture, we have very big table with the growth factors that we should change, that we supposed to that these are effect to the culture of the organoid based on the organogenesis, based on the literature, and we try to put less and high amount of this growth factor. Maybe it's better to say that we are playing with the growth factors and checking the mutation, the structure, and heterogeneity of the organoid. At the end, we have some growth factors that we can define that this growth factor with this concentration is good for culturing this type of the organoid. That's why that bladder organoid culture and set up the culturing of bladder organoid for me take 2 years here, and it's a very time consuming and
Okay.
yeah, but it's
Yeah.
just
All right. The next question is, would you tell us the specific difference of the cultural environment of organoids?
which question? Sorry, your voice.
It's one before the last. So would you tell us the specific difference of the cultural environment of organoids? So I'm assuming that they're asking for the culture media that you're using in the organoid production. What are the specific differences versus 2D culture?
In 2D culture, we use FPS, yes, and the FPS is a mixing of everything. But in the organoid, we just target some signaling pathways that we want to keep the stemness, and also we want to differentiate them a little bit. We want to have a multi-layer with different type of the cell in this structure. We want to have a structure that can recapitulate the organ, but in the cell lines, just we use FPS to culture them. Then in the FPS we cannot control everything, because in FPS we have everything and we don't know which pathway we are
Targeting.
exactly.
Right. And one last question that came up. Oh, one more. And I would ask you to please put your email address at the bottom of the page, so whoever wants to ask you questions later on or reach you, they can.
Yeah.
So they're asking if what are the strategies that you're using to avoid so when you're extracting your cells from the
Yeah.
patient, what are the strategies that you use to avoid any pollution or pollutants in your cell culture?
Yeah, it's very, very good question and very big problem in culturing of the organoids. Um, uh, Honestly, we try to use very power antibiotic like pyrimocin in our culture system, and before starting to culturing we try to wash them a lot with when we resect the tissue, we put the tissue in the media with antibiotic, and when we transfer them, the tissue is in the media with 1% of very with pyrimocin. And then when we want to work, we try to wash them with these with media with antibiotic with pyrimocin, not penestrep or streptomycin. Pyrimocin is powerful is good antibiotic for organoid culture. So yeah, in this way we until now we could you know keep the organoid and we can culture the organoid without infection.
All right. So and there's a correction to that question. He says that he wants to know how you're avoiding how you differentiating the healthy cells from the cancer cells.
Differentiate healthy from cancer?
Yeah, when you're extracting cells from the patient.
We don't extract the healthy from I understand. We try to resect the one section of bladder, for example, from tumor tissue, and the one section for normal section that it's far from of the tumor section,
Okay.
it's far
Okay.
from, and then
Yeah.
this is the
Can we embed one organoid with paraffin for IHC?
honestly, IHC is a little bit hard to doing for organoid because of the BME. When we have BME, after adding the antibody, we have a lot of background. We couldn't do that, but I established a new strategy for staining of the organoid with in a slide chamber for IF, not IHC. IHC, it's a little bit hard. It is possible, but because of the background, I don't like to do
You
that.
don't recommend it.
Yeah.
All right. I have a question. You didn't talk about limitations of this technology. Specifically, I understand there are limitations to how big the organoid can get, um, where um, um, where I believe 3 millimeter is a standard rate. What are the measures that your laboratory has taken to overcome this limitation?
The limitation of organoid technology is its cost. It's very expensive technology, and most of the lab cannot do that because all the growth factors, all the extracellular matrix, everything is expensive, and this is a big things. And also in some of the patient, when we want to do personalized medicine, especially because organoid growth rate is very slow, and it take time to have good organoids. So for some type of the patient, or for some type of the disease, maybe, it's a little bit hard to have a quick response.
Okay.
This is
Yeah,
limitation of
but what did you what about the size limitation?
uh,
Because you cannot get an organoid bigger than a few
yeah.
millimeter, right?
Yeah, yeah, the size, we have size limitation because when the organoid size is bigger than, for example, 200 micrometer, they in the middle of the organoid, the cell is start to dying. So it depends, it patient to patient, it's completely different. And also when we are working in different tissue, it is also completely different. It depends on tissue and patient, but we cannot grow them as a very big structure. Just 250 for bladder is good, but bigger than.
Okay. All right. So there's also a question about biobanking.
Uh-huh.
And so they're asking what are the applications of organoids in biobanking, and there's a question about electrical field stimulation, and this would be the last two questions, and I would ask you again to send us your slides, so I can share it with your with my colleagues, and also your email address,
Yeah.
please, at the bottom of the chat. So first question would be about biobanking and its applications, and the second one would be the electrical field stimulation in organoid culture.
Yeah. About the biobanking, after for every patient, we have information, clinical information, yes, and then we try to have a organoid of them. Then we characterize them. Then we can biobank them. It means that we can put in the freezing media like cell lines, and we can put in liquid nitrogen, and we can keep them for more than 1 year. Then we access to, for example, 100 patients at the same time, that we can work on that ones, and we can use their information whenever we need. We can thaw vial, and we can use that organoid that we know the information of the patient, we know the information about the mutation of that patient, everything, and we can use it. And yes, in the Netherlands, Hans Clever's Institute is a hub, and they are biobanking the organoids, and for some of the company. We are also here. We have a biobank of bladder organoids, and 150 around 150 biobank of bladder organoids that my supervisors talk with some of the company, and they can some of the company can use this biobank. Yeah, they are using.
Is that a fire alarm? We're hearing an alarm.
Maybe it's in the lab, but I don't know what is it.
Okay. There are many other questions that are coming up. I don't know if you have another maybe 5 minutes to answer all these questions. One is about the electrical field stimulation. The other one is about limitations of the tissue. So if you want to answer these, if you have time, 5 more minutes, you can answer these questions. If not, I'll ask everyone to kindly send you their questions via email.
For the question about the electrical field, honestly, I don't know anything. For limitation for choosing the tissue, it depends on your object, your studying objective. I don't, maybe, for example, for us at the first, we wanted to use to work on cisplatin gemcitabine resistance one, so we just choose that patient. But for the another things, no, I think no, we don't have any other limitation. How we can claim that mutation? Yeah. Organoids should be similar to original tissue. Okay, we can collect the organoid, isolate DNA, and then we can do DNA sequencing, and then we can compare this result with the result of original tissue. And they should be same, otherwise we cannot use in experiment. And the normal organoids, of course, they should be wild type. We shouldn't have any mutation. In tumor ones, we should have same mutation that we detected previously in tumor tissue. But for bladder also, there is another a little bit easier way to detect the mutation. It is a snapshot analysis that we just select the 3 most important mutation that is in the bladder. And then we try to detect that there is this mutation in the bladder, in the tumor organoids or not. If not, then we go to the sequencing, we check the sequencing, or we check the copy number variation. And how many organoids should be in each wells? It depends on tissue, but for 48 well plate, I can culture 100 organoid around. but you can also add more, because when the confluency of the organoid in each well is high, then these cells also secrete some growth factor, and these growth factors can help to have a good organoid. And this is the things that, but if they are very high, this is also, it's not good, because because of the shortage of the growth factors for them. And about the biobanking, that how we can biobank them, there is some freezing media. We can also freeze them like cell line, 50% FBS plus 10% DMSO plus 40% media. But also there is some freezing media that you can buy them, that these are safer than our mixing. You can just digest the organoid as a single cell. Then you can add in this freezing media and put in the liquid nitrogen. Can organoids provide a platform for testing vaccine? Yes, of course. Some of the groups in the Netherlands, yeah, they are working on this one, and also in the US, I think they are working, and they use the COVID-19 on liver, on lung organoids, and or some of the vaccines. They are working on this section. Yeah.
All right. I believe Dr. Kamberi wished you a very successful continuation, and thanked you for your nice presentation. So if you could please type in your email address as you
Yeah.
just did, I'm going to pin this. And so if you have any questions, you can email Maryam. She'll be responsive. And if you could please send me your slides as well, so I can share
I
it with
sent
my.
you, yeah. I sent.
I'm not sure if you have.
Okay, I again try.
All right. All right.
Yeah.
All right. Thank you very much.
Thank
And this
you.
has been another webinar, and hopefully we'll have another next month. And thank you very much for this nice presentation. And God bless.
You're welcome. Thank you for giving me
Stay
this opportunity.
safe.
Thank you. Thanks.